Okie dokie. All right, so this is just a recap from last week. There's really not a whole lot of new stuff here. So we started, I do not have chapter 1 material on this recap. I at this point hope that you guys are very comfortable with that material as far as basic terminology and anatomical terms. So this is primarily chapter 2. So all about chemistry. We said about 95% of what goes into life comes down to 4 elements: carbon, oxygen, hydrogen, nitrogen. And then you sprinkle in some metals and some a few other atoms, and you have everything you need for life. And we said that matter is space, something that occupies space and has mass. And we said that the fundamental stable unit of matter is the atom or the element. And we talked a little bit about the periodic table of elements. We talked about what the periods are as far as how the shells of energy that we have, the electrons. And then we also talked about the columns or the groups, which were how many valence electrons there were. And that's helpful because that's really what we care about for most of chemistry. We said the general structure of the atom is you have large or high mass particles in the center, or what we call the nucleus, that are positively charged, called protons, and you have neutral, or these particles that don't carry any charge, called neutrons. And then in shells of energy around the nucleus, we have the electrons. And we said that for the most part, most of the relevant elements or atoms that we deal with obey the duet and octet rules. And that is to say that atoms are either happy when they have 2 or 8 electrons in their valence shell. Hydrogen and helium are the duets, and then pretty much everything else, okay, beyond that's going to be octet rule. Just a quick note on the term isotope. I don't think this is something I specifically talked about. You can have different isotopes for different elements. For example, hydrogen. 90+% of the hydrogen that we find is in one form, and that is a form that has 1 proton and 1 electron. An isotope is something that has the same number of protons, okay, because remember, the proton is like the fingerprint for an element, okay, so if it has 2 protons, it's always helium. 1 proton is always hydrogen, etc. But you can have the same number of protons and different number of neutrons, okay, and that's what an isotope is, okay. So you could have hydrogen, for example. Instead of not having any neutrons, you could have a hydrogen with a proton and a neutron, okay? And so that would be heavy hydrogen, right? That'd be hydrogen as much heavier than the normal hydrogen that we find with just the 1 proton, right? And that would be an isotope of hydrogen, or what we call deuterium. There's also a heavier isotope of hydrogen, which is 1 proton and 2 neutrons. So it's extremely heavy, and that's what we call tritium. And tritium is sometimes used in watches and in gun sights because tritium, because that nucleus is kind of large and heavy, it becomes unstable and it breaks down. And when it breaks down, it emits light and it glows. And so you can have like really high quality watches that have this tritium in it. And that's all tritium is. Tritium is an isotope of hydrogen. You guys have heard of carbon 12 and carbon 14. Have you ever heard of that? What that means is that 12 or 14, they're talking about isotopes of carbon. And most elements have isotopes. Some of the isotopes are unstable and they're radioactive and they tend to decay. Okay, we talked about the octet rule and the duet rules. What comes out of them is the fact that we get chemical bonding. We talked about ionic bonding. That is the gain and loss of electrons. Okay, when something loses an electron, it becomes positively charged. We call it a cation. When something gains an electron, it becomes negatively charged, and that's what we call an anion, right? And okay, being without, cat being positive. Okay, when these things dissolve in water, when ions dissolve in water, okay, because they're charged, okay, because they can carry an electrical current, another term for them is electrolyte, okay? Electrolyte is just a fancy term that means ions dissolved in water. Okay? So when you dissolve charged particles in water, charged atoms in water, they're called electrolytes because they can carry an electrical current. The more ions you have dissolved in water, the easier it is for water to conduct electricity. Is that, you guys okay with that? The more ions you have in that. Okay? So we call them electrolytes. They're measured in something known as an milliequivalent, or MEQ. We measure them in milliequivalents per liter, and this is important in healthcare, okay, because you have normal levels of electrolytes in your blood plasma. The two most important ones that we run into are sodium and potassium, and I just threw this in there for the sake of completion. Your normal sodium level in your blood plasma is 135 to 145 milliequivalents per liter. You get much higher or much lower than that, and you start having problems with homeostasis. Likewise, the normal level of potassium in your blood plasma is 3.5 to 5 milliequivalents per liter. So clearly, the amount of potassium in your blood plasma is a lot lower than sodium, but we know that there's actually a lot of potassium that gets hidden away in the cells. Another concept that came out of that was that metals tend to lose electrons, right? Metals tend to want to give electrons up. Okay, so things like sodium, lithium, magnesium, calcium, okay? Most of the elements on the left-hand side of the periodic table of elements are metals. That's what makes a metal a metal, is it doesn't hold on to its electrons very tightly, okay? And coincidentally, that's why metals are so good conductors of electrical current. Okay, because all an electrical current is a movement, it's a flow of electrons. And because metals don't hold on to those outer electrons very tightly, it's easy to flow an electrical current through that. Non-metals, however, okay, tend to gain electrons. They tend to want to hold on to their electrons. Okay, so things like oxygen, fluorine, chlorine, those things on the right-hand side of the periodic table elements, okay, the non-metals tend to want to hold on to electrons, and they tend not to be the greatest conductors of electricity, okay, unless they're in some ionic form, of course. All right, and then that brought us to covalent bonding, and covalent bonding is not give and take of electrons, it's sharing of electrons, okay? And this typically occurs between nonmetals and nonmetals. Ionic bonding is metals and nonmetals. And then we said that there's a situation where you get unequal sharing sometimes. We call these polar covalent bonds. And we said that when a less electronegative atom is covalently bound to an electronegative atom, the electronegative atom is going to want to pull, okay, more of the electron density toward it. So I have a situation where I have oxygen and hydrogen, and more electron density is around the oxygen, so the oxygen part of this molecule becomes partially negative, and the hydrogen becomes partially positive because the oxygen's pulling the electrons away. And that led to something, a type of intermolecular force known as hydrogen bonding. And this is a force that molecules, polar molecules, can have. Hydrogen bonding is one example. There are many other types of intermolecular forces, but hydrogen bonding is very important in biology. And we said whenever we have hydrogen bound to an electronegative atom like nitrogen, oxygen, or fluorine, this sets us up for a situation where we have this hydrogen bonding. And here's just a picture of two water molecules. And we said that the hydrogen is going to be partially positive, right? And then the oxygen is going to be partially negative. And that attraction between the oxygen and hydrogen is what we call a hydrogen bond. Okay? That's what holds those water molecules together. That's what allows water to be a liquid at room temperature under normal conditions. You heat the water up though, right? As you heat the water up, those molecules start vibrating, and eventually as you heat it up hot enough, the vibrational energy, the kinetic energy that those molecules have is stronger than the hydrogen bonds that hold them together, and they fly off and they become steam, they become water vapor. Okay? And we also said that water is polar, and polar molecules are really good at dissolving other polar molecules. And so we said that that's what makes water a good solvent. Solvents dissolve solutes. Solutes are things that get dissolved in solvents. Water is a solvent, and then things like sodium and chloride, and potassium and magnesium, and somebody left their phone on. Happens all the time. Right? Get dissolved in water. So there we go. All right, and then that led us into talking about the chemical reactions, synthesis and decomposition reactions, right? Synthesis is putting stuff together. The dehydration synthesis was the one that I focused on. We'll talk about that today. Decomposition is breaking apart, and hydrolysis is a major mechanism. And we said, in general, anabolism is building stuff, catabolism is breaking stuff down, and the sum of all the chemical reactions of the body involving anabolism and catabolism is what we call metabolism, right? So metabolism is both building and breaking down. Another concept that falls out of this is something known as a redox reaction. It's a word that I may use from time to time in this class. And redox just stands for reduction oxidation. Okay? And if something becomes reduced, okay, when I reduce something, that means one of two things. That means I've either added hydrogen to it, or I've added an electron to it. And I just think of its charge is becoming negative. Its charge is reduced, is becoming, I don't think of reduction, but I think of negative. Oxidation, however, if something becomes oxidized, that means I've either taken electrons away or I've added oxygen to it. Okay? So these redox reactions can be synthesis or decomposition. It's a pretty broad category. Okay, that led us into talking about acids and bases. Remember, acids donate hydrogen ions or protons, same thing, right? Bases accept. One of the big bases or buffers that we have in the human body is bicarbonate, right? HCO3, or sometimes it's associated with sodium, and we call it NaHCO3, or sodium bicarbonate. That's a common buffer. Okay, it's in our blood, in our plasma, and it helps to buffer acids that are produced. Buffer those hydrogen ions because it can accept hydrogen ions. And then I just gave you an example of hydrogen chloride or hydrochloric acid, and when you put that into water, it dissociates into hydrogen ions, chloride ions, and it is the fact that it dissociates into these hydrogen ions that makes it acidic. We talked about the pH scales and the negative log, the base 10 of the hydrogen ion concentration in moles per liter, right? It goes from 0 to 14, 7 neutral. Okay, the lower we go, the more acidic, the higher we go, the more basic. And normal human pH is 7.35 to 7.45, so it's slightly alkalotic. Okay? And then that brought us into talking about the macromolecules of life. We talked about carbohydrates. We said that they kind of have a ring-like they kind of have a ring-like structure. That structure can be opened up, and you can see in its open structure. There's a nice picture of your textbook that compares the open structure to the ring structure. Okay, these are primarily what? Carbons, hydrogens, and oxygens. Okay, a single carbohydrate is what we call a monosaccharide. Saccharide is another term that means carbohydrate. Okay, an example of a monosaccharide would be glucose. Okay, that's the major fuel for our body, right, is glucose. So when we eat plant, the sugars, the carbohydrates in plants, are those sugars monosaccharides for the most part? Or are they more complex? They're more complex. They are often what we call polysaccharides, that is, multiple sugar molecules bound together. And so what happens is we have to break those polysaccharides down, okay, during digestion. We have to break them down into the monosaccharides, into glucose, right? So starch, for example, is very common in plants. It's a very, very complex polymer. Polymer is just a word that means a lot of different molecules combined together. And we have to break it down, right? And I told you guys about putting some bread in your mouth and chewing it up, and it tends to get sweeter and sweeter and sweeter as the amylase, right, as the amylase, which is an enzyme in your saliva, begins breaking those polysaccharides in that bread down into monosaccharides. It tastes sweeter and sweeter. Okay, we talked about lipids, right? We said that lipids include things like fatty acids, right? Very long chains of carbon and hydrogen. They typically are attached to a glycerol molecule here, okay? You have a glycerol molecule and then 3 long chains of fatty acids that come off of that, and that's what's known as a triglyceride, okay? This is glycerol, and then you have 3 fatty acids attached to that glycerol. And then we can take and we can modify those fatty acids and do things to them. One of the major modifications we can do is we can turn from glycerol, or from our triglycerides, into phospholipids, where I replace the glycerol with a phosphate, right, a phosphorus covalently bound to 4 oxygens, and then I have 2 lipid tails that come off of that. And where are phospholipids important? The cell membrane, right? These compose a big part of the cell membrane. Lipids are also important when it comes to cholesterol. Cholesterol is a type of lipid or fat. Cholesterol is within the phospholipid bilayer, and it makes the cell membrane kind of fluid and gives it some fluidity so it can move around a little easier, okay? And we also use lipids, fats, fatty acids, etc., to produce certain types of hormones, our steroid hormones, for example, like estrogen, testosterone, those kinds of things. We also have protein hormones, but these are steroids. And that's basically where we're at now. So what I need to do today is I need to finish talking about the major macromolecules. So I'm going to talk about proteins, I'm going to talk about nucleic acids, and then I'm going to talk about high energy phosphate molecules, ATP being the big one. And then that will segue into cytology, which it should be fairly easy because we covered a big component of the material that we needed to in lab. So there we go. You guys doing okay?
I have a quick question.
Yeah, go right ahead.
What is the difference between dissociation and dissolving, or is there a difference?
Well, really, it's dissociate is when, for example, the sodium and the chloride, that's when they're torn apart, right? When you put them in water and they dissociate, the sodium is attracted, because it's positive, it's attracted to the oxygen part of the water molecule, and then the chloride is attracted to the hydrogen part, and the dissociation is the molecule breaking down, and because of that dissociation, it is able to dissolve. So it's pretty much the same thing. Yeah, it gets dissolved because of that dissociation, but yeah, if you looked at them as basically the same thing, you more or less be okay. So, yeah. All right, well, let's go ahead and roll right into this then. Okay, so what I want to do is I now want to talk about proteins. Proteins are super important, right? We talked about many of the things they can do, right? They can act as pumps. They can pump ions in and out of our cells. They can make ion gradients for certain cells. They help catalyze chemical reactions. They make chemical reactions more efficient. And those types of proteins that do that are called enzymes, right? And enzymes, how do you know something is an enzyme? Well, if it ends with an ASE, okay, there's a good bet that if the name of it ends with an ASE, that it's an enzyme, okay? So amylase, okay, ASE, and generally, okay, somewhere in the name of the enzyme gives you some information about what it might do. So lipase, for example, lip would be a lipid, and ACE is an enzyme so it's a lipid enzyme, and so you can kind of look at it and go, oh, it has something to do with fats. I bet it helps break fats down, right? So lipase, protease. Prot is protein, ACE is enzyme. Oh, it's an enzyme that breaks down proteins. So does that kind of make sense? So yeah, right? They are important as far as anchoring, right? Cell anchoring molecules are often protein. that hold individual cells together, signaling molecules that come off of cells so they can signal each other, receptors, okay, receptors that respond to changes in the internal and external environment, okay, so many, many important things involved with proteins. So, let's talk about proteins. Proteins, okay, are made up of what are called amino acids. Okay? An amino acid is the fundamental molecular building block of a protein. Okay? Amino acids are combined together to produce what are called polypeptides. Okay? And then once I have enough peptides, polypeptides, once my polypeptide gets long enough, it begins to twist itself into a characteristic shape, and that's what we call a protein, and we'll talk about that in a little bit of detail. So, let's start at amino acids. So, in the human body, there are about 20 different amino acids. Okay? You're not going to have to memorize all the amino acids for this class, so don't. Okay? Now, you may, if you take chem, right? If you take like Chem 110, depending on your instructor, she or he may or may not make you memorize some of them. I'm not going to make you memorize them. But you have about 20 of them. Now, some of the amino acids your body can produce from scratch. Okay, there's some carbon, there's some oxygen, some hydrogen, some nitrogen. Your body can produce them. There are some amino acids that your body cannot produce, okay? And the only way that you can get these amino acids is through your diet. And these amino acids that your body cannot produce are known as essential amino acids, okay? And you guys may have heard that, right? Oh, anybody into like bodybuilding or something like that, oh, I need my essential amino acids. Well, that you do need them because your body can't make them under most circumstances. Okay, and then your I believe there are, 10 or 11 essential amino acids, and then, actually 9 or 10, excuse me, essential, and then the rest are non-essential, okay? You're not going to have to, again, we're not going to memorize them individually, so we're okay. It's all right. We missed the bullet there, we dodged the bullet. So, the basic structure of an amino acid is as follows. It contains 3 important components, okay, and I've color-coded them for you. So the first important component of an amino acid is what's known as an amine group. And amine is just a word that's derived from ammonia. Ammonia, okay? And an ammonia is a nitrogen that is bound to 3 hydrogens, okay? Okay, also known as NH3. That's ammonia. And what you do, okay, to go from ammonia to an amine is you get rid of one of the hydrogens. Okay? You get rid of one of these hydrogens and attach it to something else. Okay? So do you see here, I only have 2 hydrogens attached to that nitrogen? What happened to the third hydrogen? There you go. It got replaced for a covalent bond with this carbon here. Do you see that? You guys see that? When this happens, this is what's known as a primary amine. If you were to get rid of 2 hydrogens, that would be what we call a secondary amine. 3 hydrogens, tertiary amine. So amino acids are primary amines. So I have my amine group here. It's a nitrogen and 2 hydrogens, and it comes from ammonia. Okay? I have in the center of my amino acid what's known as a central carbon or an alpha carbon. An alpha carbon. And this little fish-looking thing is just a symbol for alpha. Alpha or central. I believe your book uses the term central carbon. Okay, but alpha carbon means the same thing. What goes on here is the carbon has a hydrogen attached to it, and then it has something called an R. Okay, R in chemistry means some other chemical stuff going on. That's what R means. Okay? R, I think of it as everything else. Okay? That's what an R group is. It's everything else. Okay? And each amino acid, each of the 20 amino acids has a different R group. Some of the R groups are very simple. Some of them are very complex and lots of carbons and hydrogens and stuff. Okay? And it is the R groups that, well, we'll talk about what happens to the R groups in a little bit. They're very important when it comes to protein, but are you guys okay with the concept of an R? Okay, does that make sense why I have R there? It's everything else. It can be all kinds of different things. Okay, so you have your amine attached to your alpha carbon, and then on the end, on the other end, you have your acid. Okay, and in chemistry, remember I said this is the most common or the most relevant acid that we run into in lots of the biochemistry of the human body, and this is something known as a carboxylic acid, or COOH, you'll sometimes see. So you have a carbon, double covalently bound to an oxygen here, single covalent bond to a hydroxyl or alcohol group here. And the reason that this is an acid is, okay, depending on the conditions, okay, depending on the pH of the solution that this is dissolved in, this hydrogen can pop off. And when that hydrogen pops off, it pops off as a hydrogen ion, okay? So that electron stays here, and then the hydrogen ion can pop off like that. So that makes it an acid. So are you guys okay with why we call these things amino acids? Does that make sense? It is an amine group, okay, attached to an alpha carbon with an R group that is attached to a carboxylic acid group. You guys cool with that? All right.
What are the names of the three amines again? The primary?
Primary, secondary, and tertiary, you will not be held accountable for memorizing that in this class, though. But it is helpful, particularly if you're interested in like pharmacology and how certain drugs are metabolized. Primary amines are metabolized very differently than secondary and tertiary because this end becomes less polar. Okay, because remember, nitrogen is fairly electronegative, but the more hydrogens I substitute for other atoms, it becomes a little less polar. So there you go. But you don't have to memorize that for this class. You do have to know the general structure of an amino acid, though. All right, you guys cool with that? All right, so what we do is we have to combine amino acids together. And this is such a critical concept, okay? It is such a critical concept that there is this vernacular term, or this vernacular saying that goes around, and it is known as the central quote-unquote dogma of molecular biology. Now, I put dogma in quotations because what is one thing that does not exist in science? What does not exist in science? Does anyone know what dogma is? When I say dogma, what does that mean?
Belief.
It is a kind of belief, but what kind of belief is dogma? When I say somebody's being dogmatic, for example, or when I say, oh, quit being so dogmatic. Like the elections are coming up, right? Right? Everybody's lining up for their favorite team, right? They got that sports team mentality, right? I'm team this, I'm team that, right? And people are becoming very dogmatic in the way that they view the world. So dogma has to do with beliefs. It's a very rigid, unchanging way of looking at whatever you're talking about. That's what dogma is. It's a very rigid, unchanging way of viewing something. And in science, that doesn't exist in the sense that anything could potentially be false, right? That's a tenet. One of the tenets of the scientific method is that anything could potentially be false. And for something to be scientific, it actually has to place itself into a position where you could prove it, or disprove it rather. You can never prove anything in science. you can only disprove, okay? So you have to be able to do tests and experiments, and that's why lab is so important for you guys, right? Is to do these experiments. And what we have found time and time again is that this is the critical pathway by which cells make proteins. And so we say that it's a central dogma, and I put quotes around the dogma because if it were shown that this thing were to break down, if we were able to somehow disprove this, well, then there you go. But so far, it's just a critical thing that we see. And that is DNA, and don't worry, we'll talk about DNA today. DNA is transcribed to RNA, and then RNA is translated to protein, okay? This is a central dogma. So, what's going on here? Well, okay, during translation, okay, those ribosomes are producing the proteins, and this is how they do it. The ribosomes, okay, are told, right, I have 20 different proteins. How do I combine, or 20 different amino acids, how do I combine them to make proteins, right? How do I mix and match them? Well, the fundamental thing that goes on is I have to chemically combine them. And what did we say that one of the most important chemical synthesis reactions was? What type of synthesis reaction? Not hydrolysis. Hydrolysis is very important when it comes to breaking.
Dehydration.
There you go. Dehydration synthesis. So what we have happen is, okay, so here I have alanine. This is one of the 20. And then I have a threonine, okay? And I am going to combine alanine and threonine to make a peptide, okay? So what I do is I take the carboxylic acid part of one amino acid and line it up next to the amine group of the other amino acid. You guys see that? And what I need to do, okay, is I need to pull a water molecule out of here. I need to dehydrate this, right? And remember what goes into a water molecule? That is an H2O. So if I pull this hydrogen out, and I pull this hydroxyl out here, do I have my water? Do I have two hydrogens and one oxygen, right? That's H2O. I've got hydrogen here, oxygen here. So I pull this OH and this H out. I pull them out, okay? And then that allows the carbon to chemically bond to the nitrogen. I dehydrate it, carbon combines to the nitrogen, okay? And this is what's known as a peptide bond. You guys cool with that? It is the result of a dehydration synthesis. I pull the hydroxyl group off of the carboxyl end of one amino acid, okay? I pull hydrogen off of the amine end of another amino acid, and that allows the carbon and the nitrogen to combine. Okay, and now I have my peptide. And so what happens is I can combine multiple amino acids like that to produce what's known as a polypeptide. And you notice, as I'm adding these up, one end of the polypeptide chain is always going to have a nitrogen, right? Right here. And then the other end is always going to have the carbon. And when we start talking about these chains, these polypeptides, there's a terminology for, well, it's near, you may have heard this, it's near the N-terminus. Whenever I say N-terminus, what I mean is I'm talking about near the nitrogen end of the polypeptide, right? Or you may hear the term C-terminus, and this is near the carbon end of the polypeptide. Does that make sense? Okay, right? Because one side of my amino acid has a nitrogen, one side has a carbon, and even if I combine the carbon to the nitrogen, I still have a nitrogen end and a carbon end. So an N-terminus and a C-terminus. You guys okay with that? So far so good?
Do you mind explaining the dehydration synthesis process one more time?
Yeah. So I have, so it's a chemical reaction where I pull the OH and the H out, right? I pull them out. You guys okay with that? And they can form water, H2O. I dehydrate this, right? And when I pull them out, that leaves an opening, right? That leaves a carbon and a nitrogen without their octet rules met. And so what they do is the carbon combines with the nitrogen, covalently bonds with it to meet the octet of the carbon and the nitrogen.
And then that's how the peptide is.
And that is a peptide bond there.
Okay.
Yeah. Does that make sense?
Yeah.
Cool.
Yeah.
Now, no problem. Obviously, enzymes are involved here, right? Because, you know, there's some energy and, you know, it's chemical reaction, so you've got enzymes that are involved, but that's what we do. And so what we do is we start adding all of these different amino acids, and you get this long chain, this long polypeptide chain. Okay, you guys cool with that? And it is this long polypeptide chain held together by peptide bonds which produces the first level of structure of a protein, and that's what we call the primary structure. Okay, proteins have several levels of structure. The primary level is the peptide bond. It is the individual amino acids being combined. And what tells the body how to combine the different amino acids? What's the information? What if the body needs to make a receptor, right? How does it know? What tells it? Oh, you need 10 of these, and 5 of these, and 4 of these, and 2 of these, and that'll link your channel. How does it know to do that? There you go. The DNA. The DNA. Yeah, remember the DNA is copied into RNA, and it is the RNA as it is fed into the ribosome. The RNA tells the ribosome, okay, you need to combine alanine and threonine and this and that. Okay, but here's the really crazy thing that happens with these proteins. So as I add these on, we can't forget about those R groups, right? This alanine is pretty simple. Its R group is just a methyl or a CH3 group. So this is an example of a nonpolar group. Okay. Threonine, however, has a bit more complicated R group. It's a CH, which is bound to a methyl and a hydroxyl group. The hydroxyl group is a little more polar. Okay. And as you add these, what happens is these R groups, all of the atoms on the R groups start interacting. You have intermolecular interactions. So some of the R groups are nonpolar, so they're going to want to get away from water, right? So maybe this is nonpolar and it's wanting to get away from the water, and this one is polar and it's wanting to move toward the water, right? And what you get these intermolecular interactions, and this long, thin line starts to twist due to the intermolecular interactions of those R groups. The primary thing that causes the initial twisting of the protein are hydrogen bonds. Hydrogen bonding is what produces the secondary structure. The primary structure is the peptide bond. All right. The secondary structure is primarily hydrogen bonding. Okay. You guys cool with that? Okay. And then as I add more and more and more on, there are more and more complicated interactions that can occur between these R groups, disulfide bonds, hydrophilic interactions. Okay, there are just lots and lots of complex interactions that can occur. And eventually that protein will fold into its tertiary structure. Okay, its tertiary structure. And this is the result of other more complex intermolecular forces. or intermolecular interactions. Alright? You guys okay with that? And then I can even have what's known as quaternary structure, where I may take different proteins and combine the different proteins together to make a very complex protein.
That's different peptide chains bonded together.
Yeah, so I may have like one, and I'll give you an example to give you a flavor for this. An example of this is a molecule called hemoglobin. And hemoglobin is the protein that transports oxygen in our red blood cells. And the hemoglobin is actually made up of 4 globulins, or 4 different proteins. And so what you have is you have a really complex globulin here, complex globulin here, here, and here. And then those 4 globulins, or subunits, 2 of them are called alpha, 2 of them are called beta, come together to form the whole protein that is hemoglobin. Does that make sense? And then these 4 proteins, also have what are called prosthetic groups, okay? And these are non-protein groups, okay, that often contain what are known as cofactors. And you guys, you read about cofactors in your reading? Well, if you did your reading, that was a word that was in there. And a cofactor, okay, is some other molecule that helps the protein do whatever its job is. Oftentimes these cofactors are things like vitamins. Like vitamins are really important cofactors. Another very important cofactor is going to be some sort of metal. Iron tends to be a very important cofactor in a lot of these enzymes. Okay? Iron. For example, hemoglobin contains iron. Other enzymes contain iron as well. Zinc is another cofactor. Okay? So you have these cofactors that come into play as well in this quaternary structure. Okay. You guys okay with that? So you have primary structure, the peptide bonding. The secondary structure begins to fold. And there are basically two general types of folding that we run into with proteins. Okay? The protein can fold in 1 of 2 configurations. You can get the protein kind of fold like this. Okay, so just kind of some basic folding, and that's called a beta sheet. Or you can get a helical type of folding, okay, and that's known as an alpha helix. So the secondary structure is often either a beta sheet or an alpha helix, and then the tertiary structure is a combination of helices and sheets to produce that globulin. And then if you have quaternary structure, then that combines with other ones to make the total protein. Okay. You guys okay with that? So here's an interesting thing about proteins. Yes, sir?
What's the coil?
Alpha helix. Alpha helix and a beta sheet. Yeah. Sorry, you guys cool with that? Okay with that? Don't be afraid to ask me questions. I'm not violent, am I? All right? I don't think so. All right, you guys cool with that? Make sense? All right. Now, protein folding, how this happens is mind-blowingly complicated, and there are actually many unresolved questions in biology, in biochemistry, molecular biology that revolve around this. How do they fold into this really complex structure? A lot of work being done right now. But what we know is, for a protein to function properly, it has to have a very particular or very specific shape. Okay? If it doesn't have that particular shape, it will not function. And the special shape that a protein has, in order for it to function, is what's known as its conformation. Its conformation. Okay? And proteins can have multiple conformations, okay? Like a protein channel, one conformation may be open, one conformation may be closed. Certain protein receptors, okay, activated and inactivated, they're different conformations, okay? But if something were to occur to change the protein shape in a way that would prevent it from functioning, that is what we call denature. When we denature proteins, we change their shape in a way where they can no longer function. This is important because if I denature enough protein in enough cells in my body, what do I do to homeostasis? I take the body out of homeostasis and potentially into a situation where we die. Right? Lots of things can cause proteins to denature. For example, pH. That's why we spent time talking about pH. pH levels that are too low or too high, or acid levels that are too high or too low, can denature proteins in our body, can change their conformation and make them work in ways that we don't want them to work or prevent them from working altogether. Too high of temperature, too low of temperature, okay? Not enough ATP, okay? Reactive molecules that are produced can denature proteins as well. Okay? You guys okay with that? Alcohols, okay? Alcohols can. In an experiment, I thought about doing this for you guys in class today, and then I held back because I wasn't quite sure what kind of trouble I'd get into. But if you guys want, and you have somebody or know somebody that has a very strong alcohol, very strong concentration of ethanol, I would recommend something like Everclear. Take Everclear, pour it into a glass, crack an egg, and put that egg in that Everclear, and watch it. And has anyone ever seen this? That egg will literally cook, quote unquote. Yeah. Because what is cooking? When I cook something. Yeah. When I cook something, okay, cooking, heat. When I add heat to something, the proteins in whatever I cook, okay, the heat, right? Heat is just kinetic energy, right? The kinetic energy of the molecules of those amino acids increases. And what do you suppose the first thing to break is? Do you think the peptide bond is going to break right away? No. Peptide bond's a tough bond. But just like water, okay, when I evaporate water, what breaks? What kind of bonds break? Hydrogen bonds, yeah. So when I heat a protein up, generally what happens is the first thing to break are those hydrogen bonds. And so I destroy the secondary structure. The beta sheets unravel, the alpha helices unravel, okay? And that's what happens when you cook something. When you cook it, right? When you cook a steak, the steak just doesn't vaporize, right? No, it doesn't vaporize. What you do is it cooks and it gets brown and it gets nice and soft and you can cut it up nice and easily. You are denaturing the protein, primarily the connective tissues, okay, of that nice juicy steak, right? I mean, it allows you to eat it. And also it denatures proteins in the bacteria that may be living in and on that steak and kills the bacteria as well. But the primary structure of those proteins is still intact. Okay, when you cook something, unless you just like vaporize it, right? You just get it really hot and totally vaporize to a chunk of carbon. But yeah, it's a secondary structure that you disrupt. You guys okay with that? Make sense? So far, so good. All right. Make sure you guys pick up your labs on the way out. Okay, so those are proteins in a nutshell. Okay. Now what we want to do is, there are actually some pretty decent pictures of that structure there. If you look on page 52, they have that primary structure. And then underneath that, they have that coiling, which is the alpha helix. And then they have kind of the crinkled sheet there. Do you see underneath there? And then the tertiary structure is right underneath that, right? And it's a combination of alpha helices and sheets. and that's a globulin there. And then finally you have a quaternary structure just to the right of that where the two globulins have come together to produce the full protein. So, you guys cool with that? All right, so let's just talk a little bit about enzymes, and I know we already did, so I won't spend a whole lot of time about this, but what fundamentally do enzymes do? Do they make chemical reactions that wouldn't occur?
They catalyze?
They catalyze or they make chemical reactions occur more efficiently, and they do that by decreasing what?
What's the action? Not the action.
You're on the right track.
Hydrogen?
No, what do they decrease? The activation energy, right? They decrease the activation energy. Remember that little graph I drew, you guys? The reactants, products, the little hill, and enzymes. And what way I think of an enzyme is, it's kind of a crude way of thinking, but I think it works. Imagine that you're at a bar, okay, and you're kind of bored. You're really bored at this bar, and there are two dudes, and they're just drunk as skunks, okay? Just drunk, really, really intoxicated. And they start getting aggressive, and you're like, oh, I want them to fight because that would be less boring, right? So the two dudes coming together to fight is a molecule that you want to create, okay? Look at that as a molecule, okay? But here's a problem. Those two dudes are drunk, and so what are they going to do? They're going to stagger, and they're not going to be able to connect. Kind of like molecules randomly floating around in your body, right? For the molecules to have some sort of, atoms have sort of a reaction, they need to be able to connect, right? So you have these drunk things floating around, so what do? Well, you bring somebody in, right? You bring somebody in to make sure, fight, and you push them in the right direction so they can connect and the fight can happen and everybody's happy, right? That is very much like an enzyme, okay? An enzyme is really good at bringing things together. Okay, making that less random, okay? Making it less random. So there's a nice little picture where you have what are called your substrates. And your substrates are your molecules or your atoms that you want to come together to combine. Or in some cases, you might want to break something down, okay? And so what happens is the enzyme, the shape of that enzyme, complements, so those atoms or those molecules can fit into it, and that it brings those atoms or molecules into close contact with each other so they can connect, right? So it takes that randomness out of there. They connect, and they form what's called a product, and then that enzyme lets the product go. Does that make sense? That's how enzymes catalyze these reactions and make them more. And I think I already talked about cofactors. Many enzymes require these cofactors. A lot of cases they're vitamins, minerals, metals, those kinds of things. And then it just talks about like glycoproteins and proteoglycans. So these are proteins that have what attached to them, if you had to imagine. What's glyco? That kind of sounds like yeah, it kind of sounds like a glucose, sugar. Oh, these are proteins that have a carbohydrate attached to them as well. Okay? So you have a carbohydrate attached to a protein, and this is generally, there's some specialized function. One area where we see this is actually mucus. I think the book actually talks a little bit about mucus. Another area where we see this is actually in blood clotting. Your platelets actually have glycoprotein receptors on them. And when these receptors are activated, it allows the proteins, the platelets to kind of clump together and initiate clotting. And if somebody's having a heart attack, a certain type of heart attack, where we don't want clots to form, we can actually administer a kind of medicine called a glycoprotein inhibitor, which inhibits those glycoprotein receptors on the platelets and prevents the platelets from what we call aggregating or coming together to form the clot. Okay, so those of you that are like looking at nursing or some field where you're administering medications, the glycoprotein inhibitors are important for people having heart attacks, certain types of heart attacks. Okay, so those are just a couple of examples. Okay, what I'm going to do is I'm going to cut off here. I'm going to give you guys, let's see here, take about 7 minutes. I'll see you guys back at 6:30. We will talk about nucleic acids and then roll right into cytology.